Okay, good morning. I think I'm going to start because we have a lot of material to cover. But before that, I got few concerns from students, so I'm just probably going to answer it before we start. In lecture 3, on the last section, when we talk about penicillin binding protein, the last statement on, I think, slide number 10 say that penicillin binding protein target for its beta-lactamase, and then some confusion happened. So if I'm talking about a target for its beta-lactamase, because this is an enzyme produced by the bacteria, which will be targeted by the antibiotic. Or we can say the sentence in another word by saying the penicillin binding protein is targeted by beta-lactam antibiotic. Okay? Confusion is clear? Okay, the second question was from one of your colleagues saying that when you mentioned the atypical bacteria, you were saying that the acid-fast bacilli. No. I said atypical bacteria is a group of bacteria. They have special characters within the cell wall that make them not possible to stain with gram stain. An example, we talk about bacteria have mycolic acid in the membrane on the cell wall. This is why it will not take the gram stain. And in that case, they will be stained by by acid-fast staining. Another example is what is the mycoplasma, and what makes the mycoplasma under these categories of atypical bacteria, because they don't have a cell wall. And then we talk about chlamydia as an example. So acid-fast bacilli is Mycobacterium tuberculosis example. For bacteria have a special lipid, or mycolic acid around the membrane. Okay? So hopefully this is clear your confusion. We start the new lecture. Okay, so today we'll talk about bacteria metabolism and growth. There is a lot of information I'm very sure that you're aware of, because this is like your basic knowledge. And when we talk about bacteria growth and metabolism, I want you to take in consideration we already cover all the basic knowledge we need for bacterial structure and function. So we wanted here today to explain what's metabolism and discuss the stages of bacterial growth. So talking about bacterial metabolism, we want to talk about how bacteria will get the energy to running different activities. And stage of bacterial growth curve, because bacteria will go through different stages, take the nutrients and then grow fast from, an area where they start to get to know the nutrients, adapt until they take them and multiply. And of course, the depletion of nutrition will make them go into like a death phase. Other thing that we'll talk about also, like how bacteria will use oxygen, and by using oxygen, they will be put in different categories, whether they need oxygen to survive or they don't need. Oxygen is a kind of toxic to them, and amount of oxygen needed by the bacteria. So all this terminology like facultative anaerobic, aerobic, and aerobic, aerotolerant or microaerotolerant, we'll talk about it and define them during the lecture. And the last thing we'll talk about, requirements for bacterial growth and culture. So we give an example of different type of media that will be used in the lab to cultivate those pathogens. Okay? So the first thing that we would like to talk about, how we get energy. So energy is, circle of two reactions. The first one is what we call catabolism. And catabolism is breaking down the large molecules into small molecules. Okay? The second one is to take those small molecules and build them in a large complex molecules. So back, if I say catabolism, means I'm using the energy. So that's, I'm releasing energy. I'm breaking down. So all these, come with the catabolism. Catabolism is breaking down, using energy. For what? For the large molecules. Put them in small pieces in order for the bacteria, because everything we talk here is about bacteria, to use them to perform different, other metabolic activities and to get the energy they need so they survive. The other one is anabolism, which take those. So anabolism is required energy. They need energy to take this small complex or molecules to build them in a large complex molecules. Okay? So as introduction for bacterial metabolism, if I have to define what's metabolism is a series of, collections of controlled biochemical reactions. When I say controlled by biochemical reactions, because this reaction is controlled by a series of enzymes that catalyze and actually constitute the metabolic pathway. Okay, so most of the organisms, they obtain energy from nutrients. So what are those nutrients? Nutrients could be from else, could be from organic and inorganic source. And these nutrients need to be processed by chemical reaction. That's why you go to the phase biochemical reaction. So it needs to be processed in order to be used. So we cannot just, get them with the whole complex and then use them. Okay? And the chemical process to break down these molecules is actually known as metabolic pathway. So when you hear metabolic pathway, irrelevant to metabolism is the introduction of the catabolism, anabolism, and all this together is controlled by enzyme to catalyze its own molecules. And if I think of metabolic pathway, it could be performed by two, metabolic reactions. Have you heard about cellular respiration ever? And what's cellular respiration? No? Exactly. So it's what I've just talked about. So cellular respiration is a process which microbes actually use to obtain energy. And this energy could be obtained in presence of oxygen or absence of oxygen. So if the energy obtained in presence of oxygen, we call it aerobic respiration. If the energy obtained in the absence of oxygen, we call it anaerobic respiration. So if I have to define the aerobic respirations, it's breakdown of the nutrients to produce energy using electron transport chain. So we'll come soon to that when we explain the whole process. But the most important in the aerobic respiration, oxygen will be the final electron acceptor. In the anaerobic respiration, again, we break down this nutrient to be used to produce energy. But the final electron acceptor here is what? Anything other than oxygen. We're still talking about, inorganic molecules that use in respiration. Okay. There is different stages of the cellular respiration. It's glycolysis, the Krebs cycle, or we call it also Citric cycle. And the last one is the electron transport chain, which should go through oxidative phosphorylation. Why I put fermentation here? Because fermentation, I'm sorry. So fermentation is quite similar to anaerobic respiration, because in the fermentation, you don't also need oxygen. There is no oxygen. So what's the difference between anaerobic respirations and the fermentation? Because both of them, they don't use oxygen. The only difference that in fermentation, they use organic molecules. So this is the only difference between, because the process is the same. Both of them work in absence of oxygen, but in anaerobic respiration, the final electron acceptor is, any inorganic molecules, not oxygen. Okay? All right. So So this is the definition of fermentation. So fermentation, again, it's a metabolic process, produce energy from any other organic molecules in the absence of oxygen. Okay? So mainly they recycle the NADA substrate and it's partially oxidized. Okay, let us just go and think of what else we need for this, process of cellular respiration. We need an electron carrier, and those electron carrier is the NADA and NADP and FADA. So these are the terminology we hear when we explain. The whole process also goes through a reduction and oxidation. Okay, so if we want to summarize the whole process, how it happened, microbial metabolism go through 3 different stages: glycolysis, the Krebs cycle, and electron transport chain. What happened in glycolysis? You have the node concept, it's energy produced either by respirations or fermentation. Okay? Both respiration and fermentation are taking glycolysis as the first step. The end product of the glycolysis for respiration, you will have a product of pyruvates or pyruvic acid. So the pyruvates in the respiration will be the first, reactant in the second stage, which is called the Krebs cycle. In case of fermentation, the process is very short. So the final end product of fermentation is the pyruvates, and the process is shut here. So if you think of the respiration happen in the presence of oxygen, and we produce what we produce, pyruvate. Okay. Glycolysis starts from where? Start from nutrient to be breakdown. So if I think of bacteria will use, for example, glucose, because not all bacteria actually use, menthol. Example for bacteria use menthol is Staph aureus. And why I'm mentioning this here, because when we talk about the reason of study microbial metabolism, it could be a tool for identification of the bacteria. So if the bacteria will use menthol, that means in the lab, there will be a sign to tell me that the bacteria is using menthol. Okay? All right, getting to the last part of the metabolism, following the Krebs cycle, and in the Krebs cycle, the outcome here is only carbon dioxide. Okay? So it's a gas. And when you look on the lab, there is certain bacteria will produce gas. So okay, so that means the bacteria have gone through respiration, and at what stage of respiration in the Krebs cycle they produce the carbon dioxide. That's why you will see in a test tube a gas that represents, the carbon dioxide production. In the last stage, where we have energy, we have carbon dioxide, there will be the role of electron carrier, which will take electronegative molecules through the electron transport chain all the way to what? Oxygen, which will be, tied to water. So the final acceptor in the respiration is oxygen. And here where the ATP synthase will actually, help in a lot of production of energy. If I think of difference between respiration and fermentation, fermentation will produce a very little energy. It's just like 2 molecules and 2 pyruvates. The respiration could actually produce up to 34 ATP molecules. Okay, respiration happen in presence of oxygen. The oxygen could be used to catalyze carbohydrates. Those carbohydrates could be glucose, maltose, or any different of sugar. But at the same time, and this is not, I don't want you to go deep. In glycolysis, other thing could be also, digested. Lipid could be digested, fatty acid could be digested. The outcome of the first system of glycolysis will depend on the bacterial species. For example, if you have The first step of glycolysis, you have pyruvates and you have a lactobacilli. Lactobacilli will interact, produce an enzyme, react with the first product of the respirations, the pyruvates, and will produce lactic acid. We also do, like when you run, for distance, you will have your, your muscles are affecting and you get tired. So this is the lactic acid. So we produce lactic acid at this stage. So the stage of the fermentation, when the bacteria interact with the pyruvate and produce enzyme, so different bacteria will produce different enzyme, will determine the final products of the fermentation for those bacteria who survive in the absence of oxygen. I know it's kind of like a lot of information, and so, but when you look on this, summary, this is exactly the key concept, and what happened in glycolysis, what happened in the citric cycle, and what happened in the final stage of the electron transport, and the difference between fermentations and respiration. Okay, if I just look on a summary of these 3 stages, glycolysis, where you have 2 ATP will be produced, in cellular respiration, you have almost 34 ATP will be produced, and in the electron transport chain, you gather all the total of energy produced. You will have different informations in different books, or you look like searching the respiration. The total energy produced, you might find it as 36 or 38. Okay, so we stick to what we discussed here in the lab. All right, so why I should worry about bacteria metabolism? Why should I, think I need to study, bacteria metabolism? So what's the reason? Okay, great. So when I study bacteria metabolism, because I will know actually how the interactions of the bacteria with the host, us, or any other host. So this is number 1. It will also help us to understand the mechanism by which bacteria cause the disease. So it's producing those enzymes. Those enzymes could be targeting, the host cell, and as a result, you will have the outcomes of disease. And the last thing is to be able to use this as a tool of identification, and we'll soon, also when we talk about, different media, we will learn that. All right. So we need to highlight what bacteria need to grow. So we already said that the bacteria will need the nutrient to produce energy. These nutrients could be required elements. So this is very essential. They cannot survive, the important one. But they also have another element, such as the trace element, which need as a cofactor, for enzymatic reaction. So the required element, essential for life, for living, the trace element is needed as a cofactor for enzymatic reaction. There is also other factor will affect the bacterial growth, like environmental factor. Environmental factor could be what? Temperature. So range of temperature that bacteria will need to survive. The pH, whether they survive in acidic or alkaline environments, or they even survive in neutral environments. The water availability, the optimum temperature, and of course the solute concentration. Let us look on details of this. Okay, the environmental factor. Environmental factor, when we talk about temperature, the temperature actually control the speed, the growth rate. So if you have a pathogen, if you look on this graph here, you have a minimum, optimum, and maximum temperature. If I want to look on the ability of the pathogen to grow under different temperature, I have to, take a note that the growth will increase rapidly as long as the temperature increase. So if I have at a minimum temperature, you see the curve will increase to reach an optimum where I have like the most, higher growth here, and then it will decline again. So each pathogen, are controlled by what we call cardinal temperatures. What's our cardinal temperatures? Is the three characteristic of temperature that pathogen can survive within this range. So the first one is the minimum temperature. So anything below the minimum temperature, no growth will happen. So I say like the minimum growth for Staph aureus is 25. If I put the Staph aureus in 18 degrees, they will not grow. There will not be any growth. Okay? If I say the optimum temperature for the cross is 37, so at 37 degrees, I have a very fast growth. So this is what optimum temperature that's suitable to have the fastest growth of that specific pathogen. The last one is the maximum temperature, and then at the maximum temperature, anything above the maximum, no growth. Okay, let us look on the second one. So if I talk about the range of the temperature, so you have pathogen will survive between 0 to 20. We call them cyclophilic or cyclophilic microorganisms. If the organism managed to grow in a temperature between 20 to 45, we call them mesophils or mesophilic organisms. Organisms handle higher temperature, so reach like 45 to 80 centigrade, we call them thermophile. And the last group is hyperthermophiles, which can grow above like 80. So if I go to 100 and 100 and more, probably the only pathogen that could survive beyond that temperature is the pathogen able to formulate what? Shabab Shabat. So we talk about different range of temperature. We say like pathogen could actually be either cyclophilic, mesophilic, thermophilic, hyperthermophilic. And then I ask you a question. If I have a pathogen who will survive behind the maximum of 80 degrees, let us say 100 degrees, they're probably an organism who have ability to form second lecture in the spore. So in the spore, we mentioned that they will be able to survive in depleted nutrients and in like higher temperature. This is why it's not possible to get rid of them with normal sterilization methods. I mentioned that in the lecture, and they need to be killed by an autoclave because autoclave have a 121 degree in the process and steam. Okay? All right, so the pH ranging between acidic, neutral, or alkaline. And if the pathogen are growing in acidic environments, then 6, we call them acidophiles or acidophilic. In a neutrophile, which 6.8, and alkalophile is above 8. So you just get familiar with those names because this is like terminology that you will hear a lot throughout the course. All right, so other than the temperature, we want to talk about how we maintain growth of the bacteria in the lab. And for that reason, we use an artificial condition because the pathogen will grow in our body, will take all the nutrients, and then the majority of pathogen that we study, they survive at 37 degrees because this is like, our body temperature. Moving from, we maintain the nutrients when we talk about metabolism, and then we talk about what kind of pH, organisms need to survive at. We talk about they need a specific temperature. Each group of pathogen, can grow in different temperature. And then the last thing, if I want to take those, bugs and grow them in the lab, I need to facilitate a condition similar to the condition that could survive inside the body or inside the host. So I need to have something called medium. We call it cultural medium, okay? Those cultural medium could be actually in a liquid format. We call them broth, or they could be in a solid format. We call them like, solid media. So media could be in either liquid or, solid. The liquid medium, which example for that is transport medium and enriched medium. When I say transport medium from the name, what we use this media for? For transportation. Transportation of what? Transportation of bacteria from one area to another. I need to hear. To host cell. Just back. I said like we actually have all the factors that will facilitate, the best growth of this microbe according to the normal status if they grow in our body. So we actually build up, environment will facilitate this growth. Okay, but when I wanted to look on a pathogen, so how will I look on the pathogen? So you have like, sore throat and you have a fever and you go to the hospital, like, doctor, I cannot survive, I cannot, have a like higher fever. So what the doctor will do? Okay, take a sample. And in the lab, they will try to find out what caused your status. And if it's like a bacteria, and I need to do like any kind of investigation, I need to put it in a media that will keep a lot of characters without being changed. This is why we use the transport media. Okay? The enriched media is another example of, transport media, which I can just define as it's just a solid form of the liquid media, transport media. The solid media produced by what? So it's a nutrient media. It's a broth, but we add a substance called agar. And the function of the agar is solidifying the liquid. Okay? Moving more. So media, when it's solid, could be either basic, enriched, differential, selective, and we will talk about selective and differential is not like a fixed rule. So the media could be basic and rich, could be differential and selective, but those two actually have some, joint thing that will make us, classify them. The media could be differential and selective. Okay? When we actually cultivate any sample having bacteria in a solid media in the lab, we see like tiny, I don't know if you have seen that in the lab, last lab. Have you seen any bacteria on a plate? Yeah. Okay, so the tiny circle things, we call them colony forming unit. Okay? And when for example, I wanted to know the amount of the bacteria in a tube or sample, I will inoculate the bacteria in my medium, which is like agar medium, and I'll put it on a right temperature in incubator. So the bacteria is growing at 37 degrees, that's called mesophilic. So I'll put it in an incubator, adjust the temperature to that mesophilic, and I'll take it out and look on the growth on that blade. The tiny small will tell me the amount of the bacteria because I'll calculate it. And this is why when you actually have this plate with a huge growth, you need to do further steps. And we call it subculture. Okay? So the unit is called colony forming unit or CFU. All right, let's talk about those different media. The transport media is non-nutritive media. It keeps the organism viable because I might need to send my, bacteria on a sample or bacteria from one place to another place. So I need an environment will maintain this bacteria alive. So I don't want to put it on something when they reach, for example, a lab for further investigation. I will not be able to generate any results because of that. Or if I want to send a bacteria, for example, I'm doing research and I want to send bacteria somewhere, I need to make sure the bacteria will stay in a viable state. Okay? The second one, needed for this bacteria transport media, they need to have substance or they are constituted of substance, will actually prevent the growth of contaminated organisms. So this is the second one from transport media characteristic, non-nutritive, but they are keeping the pathogen or the microbes alive. They should be preventing any contamination, so they should inhibit the growth of contaminated organisms or commensal. Because why I should worry about contaminated or especially commensal? Why should I worry about commensal? Wallah, Shabab, you didn't answer this question, and I did teach you nothing. Why should I should worry about commensal? Anything, just speak to me.
Can you ask the question again?
Okay, so I feel like when I use transport media, it should keep the the pathogen viable, because this is one of the important. But also this media should prevent the growth of contaminated or commensal organisms.
So the lab people won't get infected with the bacteria.
No, you're far away. Because the reason why I'm sending this, sample, for example, in a transport media, to identify what causative, agent for specific. So if they get contaminated with commensal or get contaminated, I will actually get confused. And those commensal are not actually the causative agent of the disease that you have. True? So controlling the contaminant or the commensal will help me to reach, identification of the pathogen that caused the disease. All right? Okay, the other thing, they should prevent drying, because I want to have my specimen or sample, reaching the destinations without having any damage. And example of this medium is the Storz Emis. It's a semi-solid media, and it's used actually with charcoal on it, and the charcoal will actually maintain, all these characters which we mentioned. The one is example of a media that used for fecal transport, which is called Carrier medium. Okay, this is the transport media. The basic media is media created with broad range of it's facilitate the growth of broad range of organisms, and they don't have special nutrients. So there is nothing special, but they will be able to cultivate or growth, maintain, or give the ability of growth of wide range of microbes. Example of this, media is nutrient agar and broth. You heard about it a lot in the lab. So this is the basic or the first media we use when we start, like for example, our culture. And the uses of this media, we use it to prepare other media. So we use it to prepare enriched media. Like when we prepare the lab for you, we use a nutrient agar to prepare all the stuff that we need for demonstration. Or if you use like stock use, for identification. And the other thing, I talk about subculture. So if I have bacteria that grow in solid media and I have like a lot of growth, that will make me confused. Why? Because in that plate, I might think of seeing different feature that telling me that, well, you might have mixed organisms growing there. I'll try to take, either subculture in a solid, or take like a specific colony, which, look different, and do the subculture from the primary culture. So this is the three uses of the basic growth media. Other media is enriched media and complex. From the name, that means this media will have nutrients that are more complex. It's helpful for the growth of fastidious microbes. And some of these complex nutrients could be like, protein, lysate blood vitamins, or any growth factor, or extract, yeast, or meat. So complex means that they have all these complex nutrients available in the media. What do we use this media for? We use them to isolate the organism from sterile sites, because they have a very defined, complex media will facilitate the growth of those pathogens. Example for this media is blood agar and chocolate agar. Okay, and chocolate agar is just heated blood agar. Just remember. All right, other media is the selective media. From the name, selective mean what? Selective mean it will actually allow the growth of a specific bacteria while preventing other. In order to make the media selective, I will need, in the preparation to add what? Okay.
Specific enzyme or a specific protein.
Specific substance. This substance could be, for example, it could be antibiotic, because I know that this bacteria will be affected by X antibiotic, but the other can survive in presence of X. So adding a special inhibitory substance will make the media selective. Okay? And those could be, as I mentioned, like antibiotic dyes or chemicals, mobile salts, an example. And these media actually used to obtain sample from site of normal microbiota, which the contaminant that available, but they are not really having role in causing disease. So this is what we use as selective media. Why? Because it could tell us clearly. We're talking about normal microbiota, or we're talking about a pathogenic organism. Okay. So example of this media, we have Lowenstein Jensen media. We use it for Mycobacterium tuberculosis. And we have menthol salt agar. You remember when we talk about respiration and fermentation, we said that fermentation end product will depend on different bacteria species. If the bacteria produce a specific enzyme that will interact with the pyruvate, and we gave an example, could be tool of identification. So here, Staph aureus is a pathogen that will ferment menthol. So if I put Staph aureus in a culture plate, like selective media, I'll be able to see a change in the color, indicating the fermentation of, this pathogen to the menthol. Okay? Is clear? Or no? Okay, so other example is McConkey agar, and McConkey agar is a selective media for gram-negative. What that means, if you actually culture sample and you suspected pathogen, but you're not sure, the first thing will help you if you cultivate this sample or specimen in McConkey and you see a growth. So definitely this is a gram-negative bacteria. Why? Because gram-positive bacteria is not growing in McConkey agar. Okay? And the last thing, there is a media called thiosulfate citrate bile salt sucrose agar. Okay. With all this connectivity, I'm not asking you to memorize all those names, because when you go to the lab, there will be more chance to get familiar with them and get the name things out. And this is used for Vibro, species. Okay. The other media is the rich media. It's kind of like a fluid medium. And this fluid medium designed in a special way to suppress the growth of some microorganisms and, stop the other. So similar to the selective media, it's just a fluid, and they use for specific purpose, like identifications of, infections, or Vibrio cholera, salmonella in the food and water. All right. As I said, we have selective and differential, and selective allow the growth of some organisms, inhibit the other. We talk about allow gram-negative against gram-positive, so gram-positive will not grow in that McConkey as example. Here, the differential media is differentiate between specific characters for that specific pathogen. That means what? The bacteria will actually be recognized based on the change in the color of the colony. Okay? Or a feature will appear in the plate will tell me I have to classify the bacteria based on that feature. Okay, let us look on that. So If you have incorporations of dye or metabolic substance on that media, the bacteria will use these products and actually appear in different color colonies. Example for that, just to give more clear picture. So the dye you add or incorporated on that media or the metabolic substance you add will actually make the bacteria, due to metabolic reaction, appear in different color other than the original color of the media. And this is how I use it for identification. So if I take the McConkey agar here, which look like pinkish, okay? Pinkish color mean what? The first thing, McConkey agar, that means these are gram-negative bacteria. But then gram-negative bacteria, some of the gram-negative bacteria can utilize lactose. Some are not. So in order for me to differentiate between bacteria, gram-negative bacteria, that utilize lactose, I'll see a pinkish growth in the McConkey. I can also see, a normal color, like, whitish color or like, yellowish color in McConkey. That's mean they are gram-negative, but they're not fermenting lactose. So you understand the concept of differential media? So it differentiates between, two species of pathogen. One can utilize, for example, this is like just an example, substance. We give example here, lactose, and some is not utilizing that, lactose. And the appearance will be in the colony color, which will be different from the original media color. Okay? Another use of differential media is the blood agar. We already talked about menthol salt agar, which used for Staph aureus that ferment menthol, and that will, give you different color. The blood agar also another differential media, which we'll talk about it very soon. Okay. As I said, there is no fixed rule when it comes to selective and differential media. So if you think of differential media and selective media, differential media could be selective media. How? Okay. You remember we said like McConkey agar, true? McConkey agar is selective for gram-negative bacteria, inhibit gram-positive bacteria. But also in the McConkey agar, just take the notes, what makes the inhibition of gram-positive bacteria is the presence of crystal violet and bile salt. Okay? So differential, because it have a neutral red, that's indicator when the pH change, they will have, the change in the color. So pinkish color colony is gram-negative ferment lactose. Or white colony is not fermenting lactose. The similar concept we just was talking about. If they are selective, they select gram-negative to gram-positive. If they are differential, they differentiate between the gram-negative bacteria, ferment lactose, appear as a pinkish color, and gram-negative bacteria, not fermenting lactose, which will be like whitish color in the colonies. Clear? Okay. So more example. I'm going to give example with the bacteria. You have an E. coli. E. coli, so this is your McConkey, okay? The first one. Well, let us start from the other side. This is like, neutral agar media, where you come to the lab and then you cultivate and you see, you see this like, growth here. You take a colony from the top one, which I mark as E. coli, okay? And then you move it to McConkey agar. You have a growth. That means what? That means you have a gram-negative. Okay. And you actually, in this gram-negative, manage to see pinkish color. That means this is a gram-negative pathogen that ferment lactose. Okay? In the other side, you have Staph aureus. Staph aureus is example of gram-positive bacteria. They grow in neutral agar, but then when you move them to McConkey, they didn't grow. Okay? If you have another gram-negative bacteria here, like Salmonella, you grow Salmonella in McConkey, they don't show any change in the color of the colony That means they are gram-negative because they grow in McConkey, but they don't ferment lactose.
Did we say that McConkey only for gram-negative?
Yes.
And why salmonella?
Why salmonella is growing worse? Certain type of gram-negative bacteria, as an example. So don't figure like the name. They can grow in McConkey without causing any change in the color. That means those pathogens, they don't really use, ferment lactose. Staph aureus is the only example for gram-positive, which will not survive in McConkey. Okay? All right. I talk about differential media for blood agar. So blood agar is, differential media because it differentiates about between 3 different factors. So some pathogens have the ability to, lyse the blood in the blood agar media completely. How I will actually identify those pathogens? I'll see like a clear, zone of inhibition of growth around the growth of the bacteria, okay? And we call this beta hemolysis. So if the pathogen will manage to do a complete hemolysis of the RBCs on that media, we call them beta hemolytic pathogens, okay? If they do partial reductions of the red blood cells, hemoglobin to metaglobin in the media, that means a partial products will be like a reddish, greenish to brownish color. We call them alpha, or alpha hemolytic pathogens. If the pathogen has no effect and still I see like, the growth here without any discolorations or zone of inhibition and no change in the color, we call them as gamma, hemolytic pathogen. Okay? So complete lysis is beta, partial lysis or reductions and change in the color is alpha, and no actions, no ability to do any lysis effect, we call them gamma. And then you will see that in the lab in the coming lecture. All right. The other thing that I want to talk connected to the bacteria using oxygen. So in the metabolism, again, we said that oxygen is important in the respiration. Part of the fermentation we have without oxygen. So that means the bacteria, when they have oxygen will be also breaking into different radicals. Those radicals could be toxic, like what? Hydrogen peroxide, hydroxyl, radical. So what bacteria do? Bacteria will produce enzyme, and those enzymes actually will be able to toxify those toxic effects. So detoxifying enzyme. Detoxifying enzyme like what? Catalase and sodium distimase. Okay? Those are actually work when they produce by bacteria to destroy the toxic radical produced as end product of oxygen. Okay, let us move. So if I have an oxygen radical, will give me oxygen will reduce oxygen radical plus hydrogen. In presence of enzyme called superoxide distimase, that will yield a hydrogen peroxide. So if the bacteria have a catalase, that will do what? Will end up to have a water and oxygen. How I test for that in the lab? If you take like a bacteria, you add hydrogen peroxide in the slide, you will see bubble. And this bubble indicator of productions of oxygen and water, because the bacteria manage by the enzyme catalase to actually neutralize the hydrogen peroxide, break it down to water and oxygen. Okay? All right. By this requirement of the oxygen, the pathogen need an oxygen, we call them aerobic. So aerobic or obligate aerobic, the bacteria that will never survive in absence of oxygen. So, period, they die. Anaerobic is the pathogen, they will survive in absence of oxygen. So what in the middle is the facultative one. The facultative one can actually survive in presence and absence. So try to connect the terminology with the name. Aerobic mean oxygen, and aerobic mean no oxygen. Facultative, they can have both. Okay, if you look on the first tube here, the first one, because the bacteria is growing on the top, that means they are obligate aerobic. They cannot survive without oxygen. The second one, where the growth is down in the bottom of the, tube, that means the bacteria are obligate anaerobic, so they don't need oxygen to survive. The second one, scattered all over, that means some of the bacteria could grow in presence and absence of oxygen. So the top is more oxygen, the bottom is like, no oxygen. So this tell you like they can grow in both status. There's other terminology like air tolerance, that bacteria can use oxygen, but the oxygen presence is not negatively affecting on the bacteria. Okay? The last one is microaerosols, which type of pathogen that can actually need a little tiny of oxygen to survive. Example of those pathogens, if I talk about aerobic, you have Pseudomonas. Anaerobic is Bacteroides. Facultative, we have E. coli or Escherichia coli. The air tolerant, we have Streptobiogen. And for microaerobic, we have Campylobacter. So again, those names, just try to get familiar with them. The more we talk about specific gram-positive, gram-negative, and those species, you will just get to know the pathogen. Okay. This is I will just explaining, in case of the aerobic bacteria where they have oxygen, by having catalase and sodium distimate, they will be able to make neutralizations. And then it goes for the rest. Okay, the last type of media is the aerobic media. And how can I make the media, anaerobic? Because the special pathogen I would like to cultivate, I wanted to take the oxygen from the media. So I'll add, substance to actually control the presence of oxygen. Example for this media is thioglycolate pros, and this media actually, control the presence of oxygen by this substance, which work as reducing agent. Okay. All right. Other thing that when we talk about anaerobic or microaerophilic growth of these pathogen could be controlled in the lab. For example, we have something called anaerobic jar, and in that anaerobic jar, it's something completely, it's like a tube locked. You add your culture media inside, but then you add, envelope which contain chemical that will release carbon dioxide, so that will, and hydrogen, sorry. And we have indicator, which this indicator will tell us like, if you actually come and you don't find any growth, but then your pathogen will need to grow in absence of oxygen, the indicator will tell you whether you're completely locked your jar and completely omit the presence of oxygen. Okay? Last thing is the pathogen need to grow in environment of carbon dioxide, so you can still create that long time. They used to have a jar and they have a candle which will reduce the carbon dioxide. But nowadays we have an incubator, and the incubator could control actually the percentage of carbon dioxide in that, inside the incubator. So this is what they use. All right. So bacterial growth. Bacteria, we mentioned that, I think in one of the lectures, that the bacteria go with the what we call binary division. And as the result, like one mother, pathogen will give two daughters. Okay? This is what we call generation of, cells. So the parent cell will give a rise to two cells. Okay? And when I talk about the generation time or doubling time of a pathogen is what is the time between the mother give the first two daughters. So if I tell you like E. coli generation or doubling time is 20 minutes. That means if I ask you, you have 5 E. coli's, cells at 20 minutes, what are you expecting to see, a total number of cells of E. coli after 1 hour? So what will you do? That was a question last year. Okay, in the exam. So you have 5 cells over an E. coli, 5, and it's 20 minutes. After 1 hour, that means the E. coli will doubling how many? 3 times, exactly. So you get the concept. So doubling time, or it's calculated based on the amount of the cells produced in each, division. Okay? All right. And the last thing is like how this happening. You add a pathogen in the media. So in the media, from the time you add the pathogen to the time where the bacteria will die, we just want to go through like, standard growth curve of the bacteria. So if you have the bacteria growing, you plot it on this logarithmic, scale, curve. The most important for me that is there is no zero because there will not possible to have like no growth, nothing. So it starts from 10 to the power 1 or 10 to the power 2. Each of these is almost 10x difference. So this is how we plot it. Okay, the growth curve of the bacteria will start with the phase called lag phase. Okay, in this lag phase, what will happen? The bacteria will just try to adapt and taste the nutrient they have. So at this stage, it's more of like starting of metabolic activity, which will be very high, but there is no division. Nothing is happening. I get used to the food, taste it like us, nice. And then I prepare the different parts, to start to do the metabolic reaction. So nothing happening in the lag phase. Nice, calm, take the food. Okay, great. I love it. And they will go to the second phase where it's called, log phase. And in the log phase, they start to fight. Okay, I love it. I want to eat as much as I can. So what will happen in this phase? And in the log phase, there will be like rapid growth of the bacteria because they get, their food and they grow and they, start to multiply because they are okay. They have the energy, the food, the energy and everything, okay? So in the log phase or expositional, we call it oxy-expositional phase, there will be like rapid growth of the bacteria, okay? At that stage, the bacteria will be more susceptible to antimicrobial. This is where the antibiotic targeting. We wanted to stop this multiplication and growth of the bacteria. So this is where the susceptibility of antibiotics with this pathogen. Okay, following this stage, we go to something called stationary phase. Okay, these guys have all the food, they fight on it, they eat it, then it's over. No food, no more food, completely depleted. So what they will do? Some of the bacteria will go in something called sporation. True? You remember when they don't have food, they want to stay and have like when you have economic problem, people will run to the shops like the Corona time. I mean, if you go in the US to the big store, you don't find napkins. You don't find hands and sanitizer. People get crazy, exactly. So they want to take stock and keep it. So this is exactly what happened in stationary phase. There will be no food, depletion of the food, no growth factor available in the media. So the bacteria will stop dividing. The amount of the growth and the death will be equal at this stage. You have amount of the death and the growth rate will be equal. Okay. This stage also connected to the variance factor because if I have the bacteria go in sporation, I will not be able to target it by antibiotics. True? So this is also where variance factor, such as spore formation, will be at that stage. And then, of course, following this stage, we'll go on to this. The majority of the bacteria will die because if they don't actually get more nutrients. So in artificial media in the lab, when you actually culture your bacteria, especially for research purpose, you add your medium, and then you will come after a couple of days, even for cell, just like cell line, you will notice a difference in the color of the medium. That will give you an indicator, a lot of nutrients have been depleted, so you need to add fresh medium. So pathogen who go in sporations, when you add a fresh medium, they will go back to what?
No.
They will go back to stationary phase and to log phase because you remember we talked sporation will go from spore forming to vegetative cell. So when you have nutrients, the calcium and will start to take out from the spore formation and all this cortex, and then they will just release and start to get the nutrient, and then they grow back to normal vegetative cell. So this is exactly what happened. If you supply more media, the pathogen will start to get nutrients. They go out of the spore forming, and they will, survive. Okay, I think why I should think of these bacteria growth and division, because the bacteria growth and division is a target for antibiotic. We talk about in the stationary, in the log phase, where multiplication is happening. We have all this, stage where the antibiotic will be, attacking bacteria. We will learn about how, bacteria need time, or like how long bacteria need. Doubling time will help us also to know the incubation period of the disease. Of course, the most important, part of it also as identification of this pathogen. So if you have for example, long doubling time in case of Mycobacterium tuberculosis. So if I think of Mycobacterium tuberculosis actually doubling time is 20 hours. So that's really a long time. And for that reason, those kind of gross media sometimes will actually help us to, identify those bacteria based on the liquid culture-based technique. And of course, this will have a big role and implications for the treatment of and the effect of the antibiotic. We are done. Okay, too much information today. I know people were loving what she's talking about. But anyway, the most important message I want you to carry, that when we talk about metabolism, you need to know that bacteria could do respiration. Respiration could be aerobic and anaerobic. This is connected to identifications of the bacteria, because when we talk about oxygen, the bacteria will use oxygen, and if they produce certain enzymes, they will destroy the toxic radical of oxygen products. Okay. Other things like bacteria will grow in different phases, how the bacteria grow in each phase and why, and different media that we use to, create a condition of the bacteria to survive in the lab, and what happened in each stage. And the last thing, different type of media that could be differential, selective. They could be differential and selective. What do we add for the media to make them, differential and selective? And of course, tool of identification of those selective media for different pathogens based on the product coming from bacteria will make change in the feature and the color of the colony, or appearance of, for example, like blood agar when they have these, hemolysis or hemolytic feature. This is the whole stuff that we talk about. Okay. Again, feel free to send me questions by email. And you have my contact. You can meet me if you have anything that you confuse that, okay?